Good morning, dear students of class 12 science. We are dealing with the chapter strategies for enhancement in food production. So we'll continue with that. So in the last class, we were discussing about the main steps in breeding. We have learned about the germplasm collection for variability, evaluation and selection of parents. Then we have learned about cross hybridization between the selected parents. And in cross hybridizations, we have also learned about the types of hybridizations. And the most important thing, we have learned about the procedure of hybridizations. And in the procedure sections, you have to mainly remember the term emasculation and bagging. Okay, so kindly go with the technique or procedures of hybridizations. So we'll continue with the steps of breeding only. So today we are going to learn about the selection and testing of superior recombinant, followed by testing, release and commercialization of new cultivars, and lastly we have got the multiplication of improved seeds. Okay, so these are the main steps of plant breeding. So germplasm we have already done, then evaluation and selection of parents we have already done, and we have also finished about the cross hybridization between selected parents. So over here you have to kindly remember the term. One time saying that you have to remember the term emasculation and bagging. So let us begin with the fourth step, that is selection and testing of superior recombinants. Okay, so selection and recombinants of superior parents or recombinants. Recombinants means the hybrid progeny which will carry the characteristics of both the parents. Okay, that is called the superior recombinants. Okay, so over here it involves the following steps. The first step is selection of hybrid plants that have the combination of desired characters. Hybrid plants must bear the parental characteristics which are desirable characteristics. Okay, so if the characters from both the parents are inherited to the offsprings or to the progeny, from that particular hybrid plants has to be selected. Okay, then let's say selfing means self-pollination. Okay, selfing the selected plants to several generations to produce uniform homozygous inbred lines. So after the selection of haploid plants, we have to go through the self-pollination. So that self-pollination will result in the creation or generation of a pure line. Okay, where the desirable characteristics will be maintained from generation to generation. So after self-pollination of cereal plants, we have to go through the cross-pollination. So crossing the selected inbred lines to produce uniform F1 generation or first filial generations, where the plants will produce the desirable quality and the quantities. Okay, and so that the seeds can also be grown directly. And lastly, we have to go for the selection in self-pollinated and cross-pollinated crops. Clear? So selection in self-pollinated crops result in the formation of a homozygous characteristics from generation to generation. Okay, it isolates the best homozygous genotypes present in the population. It will separate. Okay, we can separate the best homozygous characters present in the populations by the selection of self-pollinated crops. Now, those homozygous genotypes which are being selected and which will produce a pure line and which will represent a pure line, and their selection is called the pure line selection. Clear? So what we are doing, we are going for the self-pollination. We are going for the selection of those plants which have desirable qualities and which can produce desirable quantities. Okay, and this selection of self-pollination will isolate the homozygous genotypes present in the population. So therefore the selection of this particular homozygous plants which will maintain the pure line. Okay, and the process of their selection is known as the pure line selection. And homozygosity in such varieties is maintained by self-pollination. Okay, so in order to maintain the homozygosity, we have to go for self-pollination, and therefore all the plants in the pure lines will have the identical genotype. Clear? Then What about the selection in cross-pollination? We have just finished about the selection in self-pollination, but what will be in case of cross-pollination? So selection in cross-pollinations have more heterozygous than homozygous. Why? Because we have learned that in case of self-pollination, there will be production of more homozygosity. Right? Whatever the progeny will be produced, whatever the offspring will be produced, it will maintain homozygous condition. Clear? But when we go talk about the selection in case of cross pollination, in cross pollination there will be more heterozygous progeny than, let's say, homozygous one. Clear? Why we will find more amount of heterozygous than homozygous? Because pollen from one flower is deposited on the stigma of flowers of other plant. One more time, because in case of cross pollination, pollen from one flower is deposited on the stigma of flowers of other plant. So that's why there will be more heterozygous than homozygous. Okay, and in fact the plant will also have several genotypes, all of different characteristics. Okay, so some of these heterozygous characters or genotypes are found to be more superior than the homozygous one, which are inferior and which are eliminated during selection. Okay, so the fourth step is selection and the testing of superior recombinants. So we have to select those particular progeny or the haploplants with desirable characteristics. We have to go through the self pollination and we have to go through the cross pollination. After that, we have got the fifth step: testing, release, and commercialization of new cultivars. Okay, so the newly developed hybrid varieties, after cross pollination, self pollination, whatever the hybrid varieties are produced, all right, so we have to go for their testing, we have to go for their release, and after that we have to go for their commercialization. That means we have to release them into the market. So the newly developed hybrid varieties of plants are cultivated in research field, okay, and evaluated for their production, okay, for their quality, amount of nutritional contents on that particular plants, about the taste, color, and whereas they are raised into disease or not. Clear? So they are grown under or they are cultivated under ideal fertilizer applications, irrigations, and other crop management practices for at least 3 growing seasons at several locations, at several habitat in different climatic conditions. That is called testing. All right, and the performance of these new varieties are compared with the old varieties, and the best selected varieties are isolated and they are released by the released committee for the cultivation in a given area. Okay, and finally after that what we do, we'll go through their multiplication. But over here kindly remember the term ICAR. Go through the page number 512 and remember the word ICAR, Indian Council of Agricultural Research. Okay, ICAR. Go through the abbreviation of ICAR. So after testing, release, and commercialization of new cultivars, we'll go to the next step, which is called multiplication of improved seeds. So in plant breeding, the term seed refer to any part of the plant which is used for the growing of the crop. One more time. In plant breeding, the term seed is used to represent any part of the plant that is used for the growing of the crop. For example, like in case of sugarcane, stems, okay, in case of potato, the tuber, in case of grain, for wheat, okay. So these are the different parts of the plant which are being used for the growing of the crop. So these are known as the seeds. Clear? Now, mainly NSC, National Seed Corporation, remember this word, okay? NSC, National Seed Corporation. They will streamlines raising the seeds of the improved varieties. Clear? And there are around, let's say, 13 state corporations and 100 major seeds companies which are raising quality seeds of the improved varieties. Okay, so these seeds are free of weed seeds, they are free from pathogens and pests, they are of high order of purity and have a high rate of germinations. Now such seeds are certified and packed in a sealed package as certified seeds. Now each sealed packet carries information, a state of taste, it contains purity of seeds, absence of weeds, and presence of inert matter, special features if any, and validity of certificates. All these things are mentioned in the packet where the seeds are being sealed. Clear? So here we have finished the steps in plant breeding. So kindly go through it one by one. Okay. Now we'll see the some important high yield crop varieties. So first we'll go through the wheat. So over here you have to remember the name of the wheat. Okay, so several high yielding wheats and disease resistant wheats have been produced. Clear? So here we'll just go with the name of the wheat. Okay, you have to remember that name. So remember the varieties such as Sonalika, Kalyan Sona, okay, which were introduced all over the wheat growing belt in India. So over here, just you have to remember the name of the wheat, okay, Kalyan Sona and Sonalika, which were introduced all over the wheat growing belt of India in 1963. Then we have got wheat variety Wheatley, okay, which was introduced into India from Australia. Then Oriental dog variety from Japan, okay. Then Pusala, a variety of wheat were developed at IARI. IARI means Indian Agricultural Research Institute. Okay, by exposing Sonora 64 and Lerma Rosa 64A, the maximum drop variety of wheat to gamma ray. And Sarbati Sonara is also the variety of wheat which was developed by by M.S. Swaminathan, who is known as the father of Green Revolution in India. So among the wheat, you have to remember the name Kalyan Sona, Sonalika, Lerma Rojo, okay, 64A, Sonara, then Sonara 64, then Sarbati Sonara. These are the name of the wheat which you have to remember. These are the high yielding varieties of wheat. Okay. Then number 2, we have got the rice. So here also you have to remember the name of the rice. Okay. So rice production in India went up from 35 million tons to 89.5 million tons. So let's see the few varieties. Okay. So semi-dog varieties was developed from IR8. Remember carefully, were developed from IR8, developed at International Research Institute IRBI, Philippines, and Taisung Native 1 TN1 from Taiwan. So, what are the varieties of rice that have been developed? Remember the name Jaya and Ratna, which were developed in India. So, these are the semi-dwarf varieties of rice, okay, developed in India, Jaya and Ratna. And the new varieties of rice are Jaya and Ratna, Remi, Atomita 2, Chingua 1 and Guan 18. So these are the different varieties of rice. So kindly go through the name of the rice. Then sugarcane. Over this very important, okay. Sacharam barberry is originally grown in India, but it has poor sugar content and poor production. Listen very carefully. The Sacharam barberry, which is originally grown in India, has poor sugar content and sugar productions. Then tropical cane sugar growth in South India. The name Sacharam officinarum. Okay, this has thick stems. It has high sugar yield, but it was not able to grow in North India. Okay, so this sugar cane, Sacharam officinarum, which has grown in South India, it has thick stems, high sugar yield, but we are not able to grow in North India. So what we can do, by the help of the plant breeding technique, we can combine the genes of these two particular sugarcane varieties. Okay, so these two species were successfully crossbreed. So they went for cross pollination. So therefore, the varieties which was produced, all right, the progeny which was produced contains the desirable characteristics. And what are the characteristics? High sugar yield, okay, high sugar, high production, they got thick stems, and the ability to grow in North India. So the characteristics of both the parents were present in the progeny. And what are those characteristics? High productions, high qualities, okay, thick stems, and the most important thing, it was able to grow in North India. So by this way, we are able to grow such a sugarcane progeny which has the characteristics of both the parent, Sacharum barberry and Sacharum officinarum. Clear? Next one, millets. Remember the name? Hybrid Basra, PHB, BJ, BK, and Jowar CSH series, which has been successfully developed in India, and these varieties were resistant to water stress. Okay, the next one is maize. So protein-rich varieties of maize has been developed. Name is Proteina and Ratan. So this has got high protein content. Okay, these were developed by breeding methods, and high yielding varieties of maize such as Ganga and Deccan was also being produced in India. So kindly remember the name of the crop which has got high yielding. Okay, so remember the name of wheat. Okay, we have done wheat, we have done that rice. Okay, then sugarcane, millets, and maize. Clear? And kindly go through the page number 514. In the page number 514, you'll find the list of important research centers. So kindly go through. You'll find the CFTRI, Central Food Technology Research Institute, located in Mysore. Then CPRI, Central Potato Research Institute. Okay. Then Rice Research Institute in Cuttack. Then IRRI, International Rice Research Institute, Philippines. Then IARI, Indian Agricultural Research Institutes, located in Delhi. So kindly go through the list of these important research centers. Clear? So this much for today. In the next class, we'll learn about the plant breeding for disease resistance. We will talk about mutational breedings also. Clear? So till then, stay safe, keep on studying. See you in next class. Thank you.